Welcome to Grow with Biology. Today's topic is glycolysis. Glycolysis is a type of carbohydrate metabolism. It comes from the word glykis, which means sweet, and lysis, which means splitting. It is also known as EMP pathway. EMP stands for Embedden-Meyerhof-Parnas pathway. Now, glucose, which is composed of 6 carbon, breaks to 2 molecules of 3 carbons sugar, and that 3 carbon is pyruvate. So, one molecule of glucose will break to form 2 molecules of pyruvate, whereas glucose has 6 carbon and pyruvate has 3 carbons. It takes place in cytoplasm. It is found both in prokaryotes and eukaryotes. Now, glucose, it is stored in the form of glycogen, starch and sucrose, and glucose via pentose phosphate pathway, it can give rise to ribose 5-phosphate, and glucose, if in presence of oxygen, it goes oxidation via glycolysis to form pyruvate. We will focus on the process of glycolysis. Means from glucose we are going to form pyruvate. Now it has total 9 steps. These 9 steps is divided in 2 phases. One is preparatory phase and the second is oxidative or payoff phase. Now preparatory phase has 4 steps, step 1 to 4, where the glucose prepare itself to give rise to energy. And the second phase, that is oxidative or the payoff phase, is the actual phase where the glucose releases energy. In the preparatory phase, it consumes energy, whereas in oxidative phase, it releases energy. Oxidative phase comprises of step 5 to step 9. Now, first is the preparatory phase. Now, this preparatory phase starts with the structure of glucose. And the first step, which is headed by the enzyme hexokinase, glucose gets converted to glucose 6-phosphate. It utilizes ATP. This ATP is the energy source which glucose uses to form ADP and glucose 6-phosphate. So, glucose has been converted to glucose 6-phosphate in presence of the enzyme hexokinase and utilizing one molecule of ATP, and it which get converted to ADP. Now remember, when ATP is converted to ADP is adenosine triphosphate, it is getting converted to adenosine diphosphate. Tri converting to di means it releases one phosphate, and this phosphate is added to glucose. So glucose is now converted to, with addition of one phosphate, it has now been converted to glucose 6-phosphate. The second step is isomerization reaction. There is no change in any of the groups, only the position has been changes. It is headed by the enzyme phosphohexoisomerase. So glucose 6-phosphate forms fructose 6-phosphate. Now the third step, it is headed by enzyme phosphofructokinase 1. Remember, in the last step where glucose 6-phosphate is converted to fructose 6-phosphate, notice the arrow. It is a bidirectional arrow, which means they can be easily reconverted. But in the first step, it is unidirectional arrow. In the third step, it is also a unidirectional arrow. Means glucose will always form glucose 6-phosphate, and glucose 6-phosphate can never be converted to glucose. Or fructose 6-phosphate is never converted to fructose 1,6-bisphosphate is never converted to fructose 6-phosphate. Now looking forward to the third step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Now B specified, it is bisphosphate. Bisphosphate, if you look at the structure, you will find that to the structure of fructose there is phosphate at two position. One is position 1, another is position 6. If the phosphate is attached to a single carbon, then it will be diphosphate, but if it is present at two different position, then it is bisphosphate. The enzyme required is phosphofructokinase 1. Again, it added one energy molecule, that is ATP. So again, triphosphate is converted to diphosphate. So one phosphate is added to fructose 6 phosphate, thereby giving rise to fructose 1,6 bisphosphate. Here fructose 6 phosphate uses one ATP molecule which releases ADP, and the fructose 6 phosphate gets attached to one another phosphate group, so the resulting molecule is fructose 1,6 bisphosphate. Step 4, again an isomerization reaction, the cleavage of 6 carbon sugar phosphate to 2 3 carbon sugar phosphate. So fructose 6 phosphate, fructose is a 6 carbon sugar. It gets splitted into two structures. One is glyceraldehyde 3 phosphate, another is dihydroxyacetone phosphate. These two structures are formed in the end of preparatory phase. Now the structures dihydroxyacetone phosphate is further moved to the next phase, that is the payoff or the oxidative phase. Again we can revise: glucose converting to glucose 6 phosphate, glucose 6 phosphate converting to fructose 6 phosphate, fructose 6 phosphate converting to fructose 1,6 bisphosphate, and then fructose 1,6 bisphosphate splitting into two products, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Now dihydroxyacetone phosphate, which was formed in the first phase, moves towards the second payoff pathway and it gives rise to glyceraldehyde 3 phosphate. Now glyceraldehyde 3 phosphate, there are two molecules will be formed both from the structures, and then this glyceraldehyde 3 phosphate is converted to 1,3 bisphosphoglycerate. Again remember, we specified it is bisphosphate, means two phosphates are there but their position is different. One is at first position, another is the third position. So it is 1,3 bisphosphoglycerate. The enzyme required was glyceraldehyde 3-phospho-dehydrogenase. The enzyme is dehydrogenase, means it will remove hydrogen. When it is removing hydrogen, at that point phosphate group has been attached. You can focus to it. You can see at some positions. Now here in this position where in glyceraldehyde 3 phosphate the end group is COH, and in 1,3 bisphosphoglycerate that hydrogen has been removed and it has been substituted with phosphate. This hydrogen has been removed which has been substituted with phosphate. So it is dehydrogenase, means removal of hydrogen, and to that position there is addition of phosphate. The next step is 1,3 bisphosphoglycerate converting to 3-phosphoglycerate. Means in this position it is removing this phosphate which is highlighted, and then this phosphate is attached or taken up by the ADP molecule. Here is the first step where ADP is converting to ATP, diphosphate converting to triphosphate, means two is getting converted to 3, means there is addition of one phosphate. So the structure is 3-phosphoglycerate. Again 1,3-bisphosphoglycerate in presence of the enzyme phosphoglycerate kinase, it is getting converting to 3-phosphoglycerate. There is one addition of there is a release of one ATP molecule. Now if you can notice, you will find that everywhere it is written 2 ADP and 2 ATP, because there was two products which was generated in the first preparatory pathway, and then both the products will move towards the same pathway. So there is two products generated at the end. Now the eighth step, where the 3-phosphoglycerate is converted to 2-phosphoglycerate. Again there is change in the position of phosphate. Here it is at first position, and this first position now converted to, sorry, this is the third position, and which has been converted to second position. It is phosphoglycerate mutase. Ninth step, 2-phosphoglycerate is converting to phosphoenol pyruvate with the help of enolase, and there is removal of water molecule. Then phosphoenol pyruvate is converting to pyruvate. This is the second step where ATP is generated. The name of the enzyme is pyruvate kinase. Remember, whenever there is change in ATP or ATP or energy generated reactions, there the enzyme name is kinase. First step there was ATP used, that is hexokinase, and now in the 7th and the 10th steps there is ATP generated, again it is phosphoglycerate kinase and pyruvate kinase. Now we have three steps where reactions where ATP was used. First was in the first step it was ATP used, and in the 7th and 10th there was ATP released. Now when we sum up this whole reactions, we find that there is net gain of only 2 ATP. Now this reaction we started with glucose. Then in the first step we used two ATP molecules and then two NADH, and then by two ADP and two PI, which gives 2 pyruvate, 2 ADP, 2 NADH plus 2 H plus 4 ATP and H2O. 4 ATP, 2 from 7th step, and last steps 2 ATP. So on total 4 ATP has been generated, but 2 ATP was used in the first step. So the net reaction is gain of only 2 ATP after glycolysis. So after glycolysis, when there is formation of pyruvate, we will find 2 molecules of pyruvate is formed and 2 ATP molecule is formed. Thank you.